Welcome to my channel. Today I want to tell you guys about a technique called Thioflavin T Fluorescence Assay. This assay is used for detecting the formation of amyloid fibrils over time. When amyloid fibrils form, they're usually proteins going from an alpha helical conformation to a beta sheet conformation. The beta sheets will kind of stack and the side chains will be sticking out, and Thioflavin T will go in between these side chain groups and it will change, it will give signal. And so it's very specific to amyloid fibrils, and because of that, if you form other types of aggregates or anything that is not an amyloid fibril, you will not get a signal. So this way you know that you definitely get formation of the amyloid fibrils. To do the experiment, you need 25 microliter of THT from a 1 millimolar stock that you have to prepare, and 75 microliter of PBS, pH 7.4, and 10 micromolar fibrils or 100 micromolar of the free monomer. And if you're working with just protein, usually I do 10 microliter of a 0.3 mg per ml solution, but you can try different concentrations if you want to test different conditions. And you can also add other ions or other things to see if it will affect the amyloid formation, so there is some flexibility there. Then you have to have 96 well plates with clear bottoms, and you have to cover the plates. So once you do that, you open the software, and after you turned on the instrument, you go to protocols, you open a protocol that you think is suitable. Then you can edit that one as you like, but I usually start from some other protocol that I usually edit it. And but you can also start from scratch if you like. So my protocol has a temperature of 37 in the beginning, because that is the temperature that is best for promoting amyloid formation. Then here I'm adding a shake step because it's good for promoting amyloid formation. And then the fluorescence measurement should be as stated on here. Excitation is about 450 nanometer and emission 485 nanometer, I believe. And you just want to also make sure that you're selecting the right wells. So before you put the cover, make sure you remember which wells you're looking at, so you can select it here. Then the total runtime is pretty long, so you want to make sure that nobody wants to use this plate reader. So usually I do this experiment on the weekends, and it is about 48 hours or 72 hours long. So I select the total time, and I usually do 1 read per hour, and I had a shake step before every read. Also, you want to select the wells again, because there is one selection that shows which one to read and one that shows which one to show the data for. So there were 2 places where we had to select the wells. Then you put the plates inside. Make sure that the 96 well plate is selected, and the read is from the bottom since the cover was opaque and not clear. Then you select the green button to start the read, and it will equilibrate to 37 degrees, and then it will start reading. And once it gets to 37 degrees, the plate will come out until you click OK again, and then it will go back in. So after you do that, then you can leave it for the selected time, and yeah, that's it. Thank you.